Yes, a very warm good evening, everyone. So, once again, we are back from the Team Biopoint Stream the World of Bio with yet another session on Cell, the Unit of Life, lecture number 2. Yes, so hope you all are perfectly fine and ready for this session. Yes, Yamini, Sheikh Sanali, Sanya, Binaya, a very warm good evening, all of you. So, with the same Josh that we had yesterday, let's move on with today's session on Cell, the Unit of Life, lecture number 2. Yes, reminding you to join the Telegram group. The direct link to join is given there in the description box, and that is t.me/neat underscore biopoint. Yes, Ankit, if you want to convey anything, please try to convey in English. Okay. So do hit the like button, do share it with your friends, and do subscribe Biopoint Stream the World of Bio. So we are moving on with today's session on Cell, the Unit of Life, lecture number 2. Yes, yes, yesterday we have discussed about the case of plasmid, right? And extra say, chromosomal material inside a cell. Yes, inside cytoplasm. We have said that in case of plasmid, and yesterday Binay asked me out about that. What is about in detail about plasmid. So today we are going to start our class with the topic nucleoid. Yes, we are going to start today's topic with nucleoid, and we have plasmid right in the first slide itself. Yes, so we are moving with today's session. So, what is nucleoid? Nucleoid is formed of non-membranous naked circular genomic DNA and protein. So, what is this naked circular genomic DNA? That is, there is no membranous covering around this chromosomal material. The membranous covering around the chromosomal material is absent. Yes, and they have a single chromosome or a genetic material that is highly coiled and just placed inside within the cytoplasm without any extra membrane. Yes, yes. So many bacteria have small circular DNA or plasmid outside the genomic DNA. It gives some unique phenotypic characters, example resistance to antibiotics to bacteria. So what is this plasmid? Plasmids are certain extra chromosomal materials other than the chromosomes that is present here. Yes, other than this chromosome, we have certain extra chromosomal material in the cytoplasm. Yes, and those extra chromosomal material that is outside this cytoplasm is known as plasmid. And what is this plasmid? What is the function of this plasmid? It gives some unique characteristic, unique phenotypic characteristic to bacteria. There is certain resistance to antibiotics. Some bacteria that may be first time it may be what applicable to certain antibiotics, and later on it will get what it will acquire a certain immunity against that, right? It will not, certain antibiotic will not get acquired when we have some certain disease, we are using certain antibiotic. Some will get cured and some will not get cured. That is, some bacteria just get used on to that, and some is just uh, keep is in a modifying state. That is, it will be destroyed. It will be capable of destroying by certain antibiotics. So we can see some external observable characters. That is what is meant by phenotypic characters when this plasmid is present inside a bacteria. Is it clear? Yes. Wait, let me open the chat section. Yes. So that's what we have discussed about nucleoid and plasmid. Yes. Give me thumbs up on the screen. Yes. Someone asked me a doubt. Yummy. Yes. So this eukaryotes also contain a layer of peptidoglycan. No. Only prokaryotes has a layer of peptidoglycan. Eukaryotes, in general case, eukaryotes doesn't have a layer of peptidoglycan in their cell. Okay. Is it clear? If it's fine, we will move on with the next slide. So we have completed about nucleoid, that is a single chromosome without a membranous covering, and plasmid, that is an extra chromosomal material outside the single and single large chromosome. Okay. Yes, Binaya, Sanya, Yamni, Sheikh Sonali, Calvin. Yes. Next topic is flagella. So what is this flagella? Yes, as we all know, what is this flagella? Flagella is this thin filamentous extension from the cell wall of a motile bacteria. Yes, we can say that if flagella is present in the body of a bacteria, in most cases, if we can say, if flagella is present in case of, the cell wall of a bacteria, it will be motile, because what is the function of flagella? It is, the function of flagella is motility, right, or mobility. Yes, for locomotion, this flagella is present in their body. So we can see most of the bacteria with the presence of this flagella or any locomotory organ locomotes or are capable of moving. Yes, their number and arrangement are varied in different bacteria. Bacterial flagellum have three parts. Yes. So as you all know, in some organism they have two flagella. Two flagella. Some is uniflagellated, some is biflagellated, like that. Yes. And the case of bacterial flagellum, it has three parts. That is, it has a filament, that is the longest part. Yes, if you check here, this is the longest part. So this is known as the filament. Yes, and this portion, this green colored portion is known as the hook, and the end portion is known as the basal body. That is the bidirectional rotatory, but that is the body or the portion attached to the bacterial cell wall. That is known as the basal body. Yes. Which one, Ankit? Is this present in all prokaryotes? Which one? The filament is the longest portion and that extends from the cell surface to the outside. Yes, from the cell surface to outside, the longest part is known as, the longest portion of the bacterial flagellum is known as what? Filament. Flagella. We have said that flagella are present in the cell wall of motile bacteria. If there are non-motile bacteria, if there are non-motile prokaryotes, flagella will be absent. Yes. In case of motile bacteria, flagella is present. So that's all about flagella. So it has three main parts, and the last point, that is the filament, is the longest portion that extends from outside, from cell surface to the outside, is previous year question. Yes, Ankit. So hope that's clear. Is it clear, everyone? If it's fine, we will move on with the next topic, pili and fimbriae. Come on. If it's fine, we can move with Kylie and Fimbriae. Yes. Yes, Binaya. Sania, Ankit. Fine. Yamni and Calvin, where are you? Yes, hope you all are here. So, these are surface structures that have no role in motility. Pili and fimbriae, they are the structures, these elongations. Okay, these elongations. Look, one long elongation, and that is known as pilus, and all other elongations are known as fimbriae. Okay, so if we when we see that we just felt that, or we just think that these are useful in motility or in movability. No, their function is entirely different from that of motility. Okay, so pili or singular pilus are elongated tubular structures made up of a special protein called pilin. So the protein that is present in case of this pili is pilin protein. Okay, pilin protein is present in case of this pili. And what is the function of this pili? It functions as a sex attractant. Yes, it functions as a sex attractant. Attracts the another bacteria for conjugation or sexual needs. Yes. So the protein present in pili is pilin, and the function is it functions as a sex attractant. Yes, Yamini. So fimbriae. Fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they help to attach the bacteria to rocks in streams and to host tissues. So what is this fimbriae? Fimbriae are certain bristle-like structures that helps in the anchorage to the substratum. Helps to anchorage to the substratum. Yes, right. Yes, helps to anchorage to the substratum. And what is this anchorage? That is helps to attach to the rocks. For example, a bacteria, if it should be placed on a rod, or if it should be kept on a rock, or if it is found in a rock, we can say it is attached to the rock with the help of this small bristle-like fibers called fimbriae. Okay, and that is when it has to attach to a host tissue to this fimbriae comes into help. Yes, that's about pili and fimbriae. And the what to say? Yes, the previous slide we have read about, or we have studied about flagellin, and in that we have to study the protein present in case of flagella is flagellin. Okay, noted. In flagella, the protein in present in case of flagella is flagellin. Yes. And what is the function of flagella? The function of flagella is locomotion. Locomotion. Yes, Ankit. Okay, fine. So that's about pili and fimbriae. So we have learned about two proteins, that is protein present in flagella, flagellin, and protein present in pili, that is pilin. Okay, and the function of pili as sex attractant. Function of flagella is to help in locomotion. Function of fimbriae is to help to anchorage or help to attach to the rocks or help attach to the substratum. Substratum means the host body. Any other body. Okay. Is it fine? Is it fine? So that we can move on to the sixth point, that is about the ribosomes. Yes, everyone. Most important topic in case of cell organelles in prokaryotes. Yes, you have to have a clear focus on this topic of ribosomes. Okay. So yes, Binaya, Sania, Sheikh Sonali, everyone. Yes, they are associated with plasma membrane of prokaryotes. So ribosomes are associated with the plasma membrane of prokaryotes. They are about 15 nanometer by 20 nanometer in size. Yes, their size is 15 nanometer by 20 nanometer. They are made up of two subunits, 50S and 30S, together form the 70S prokaryotic ribosomes. Yes, before moving on with subunit, let's recap something. We have already covered that. We have already learned that. Yes, Calvin. We have already learned that the ribosomes in prokaryotes is 70s type of ribosomes. Yes, 70s type of ribosomes. Okay, the ribosomes in prokaryotes is named as 70s type of ribosome. Okay, and the ribosome in eukaryotes is named as 80s type of ribosome. Yes, and in eukaryotes it is 80s type of ribosomes. So that's about 70s type and 80s type. What does this S indicate? What does this S indicate? S indicates sedimentation unit. Okay, or a sedimentation coefficient. Yes, sedimentation coefficient or sedimentation unit or Swedberg unit. Yes, sedimentation coefficient or sedimentation unit or Swedberg unit. Oh my god, here B. Okay, not V, B. Swedberg unit. Okay. So S denotes sedimentation coefficient or sediment Swedberg unit. What is this S or sedimentation coefficient or what is this Swedberg unit? It is a measure of their density and size. Yes, measure of their density and size is known as sedimentation coefficient or Swedberg unit. Hope it's clear. Are you able to follow till now? So that we can move with subunits. So that we can move with subunits. Yes. Yes. Yes, Ankit and Calvin. Yamni, Binaya, where are you? Come on. Okay. So subunits, yes, Binaya. Okay. So if it is if it is like this, okay, a single unit, this can be said as a single unit, right? If a structure is like this, if ribosomes are like this, we can say that as a single unit. But in case of ribosomes, it is two unit. Okay, it is two units like this. Okay, one second. Oh my god. Please wait. It may take some time to draw with the mouse. Yes. Yes. Oh my god. Okay. Since the line came in between, I am just highlighting that. Okay, there is no certain structures like that. Okay, fine. So these are two subunits in case of ribosomes. Yes, two subunits in case of ribosomes. That is, we have said that in case of prokaryotic ribosome, we have 70s type of ribosome. Yes, we have said that 70s type of ribosomes is present in case of prokaryotes. So the large subunit, there will be a large subunit and a small subunit. Yes, there will be a large subunit and a small subunit. And if you are moving in case of large subunit, it will be 50s type. 50s type of ribosome. Yes, and the small subunit will be of 30s type. That is the measure of their density and size will be 30s or 30, and the measure of their density and size in case of large subunit will be 50. So both this large and small subunit together constitute this 70s type of ribosome. Don't think that on adding 50s and 30s it will be 80, so it will be 80s ribo type. It is not like that. The subunits is entirely different from that of the sum. Okay. So large subunit is 50s and small subunit is 30s. Okay. And both these subunits, both subunits are associated with the help of Mg++ ions. Yes, Mg++, that is magnesium ions, magnesium Mg2 plus ions. Okay, Mg2 plus ions helps in the association of large and small subunits. Is all about subunits clear? Is all about subunits clear? In case of 70s ribosome, large subunit is 50s type and small subunit is 30s type. Yes, and both these are associated with the help of Mg++ ions or magnesium 2 plus ions. Yes, and don't think that on adding this we get 70s type of ribosome. It's entirely different from that of the sum. Yes. So magnesium is present between, or magnesium ion is present between the large and small subunits, and that helps in their association. Yes. So hope that's perfectly fine. So if we are moving in case of yes, if we are moving in case of eukaryotes, the ribosomes are ATS type, right? The ribosomes in case of eukaryotes is ATS type. Okay. So in case of 80s ribosome, the largest subunit is 60s type and the small subunit is 40s type. Okay, both this together constitute the 80s type of ribosome. Both the large and small subunits are associated. Excuse me. Both these are associated with the help of Mg++ ions. Okay. So that's all about the introduction of that's all about these subunits. So we are gonna move with the slides. So hope that's clear. Yes. So they are made up of two subunits, 50s and 30s unit together form 70s. This refers to sedimentation coefficient, that is a measure of their density and size. So if you look here, 50s units and 30s unit together forms a 70s unit, and between this both are held together or both are associated with the help of Mg++ ions. Okay, we will not write Mg2 plus, we will write as Mg++. Okay. So we have completed nucleoid. We have completed nucleoid, flagella, pili, and fimbriae, and now we are gonna complete ribosomes. Yes, everyone. So functions of ribosome are the site of translation. What is translation? What do you mean by translation? Translation refers to protein synthesis. Protein synthesis. Yes, protein synthesis is known as translation. Yes, you have learned in your class 9 that what is the function of ribosome, that is protein synthesis. And in class 11th, we have gave a term for this protein synthesis, and that is translation. And in class 12, we are gonna give a detailed explanation. That is an entirely one topic in the chapter molecular basis of inheritance in case of class 12. Okay, translation in case of trans 12. Yes, yes, Calvin, Sanya, Vinaya, Yamni, Yamni, your chat is not at all visible for a long time. Sheikh Sonali, Ankit, yes, everyone. So we are moving. So several ribosomes may attach to a single mRNA and form a chain called polyribosome or polysome. Yes, for mRNA and all we will learn in case of what 12th topic, 12th class. Okay, but mRNA is a type of RNA. Just learn in such a way. mRNA is a single strand of RNA, type of RNA, and in that several proteins get attached. Yes, several proteins get attached, as you have seen in this figure. Several proteins get attached to that strand, and that entirely is known as what? Polysomes. Yes, that entirely is known as what? Polysomes. Ribosomes of a polysome translate mRNA into protein. So, what is the function of ribosome present in this polysome? They translate this mRNA to proteins. Yeah, translation of mRNA to proteins in case of polysomes is the function of ribosomes. Yes, everyone. Hope the functions are clear. If it's fine, we will move on with inclusion bodies. The next cell organelle, the last and final cell organelle that we are going to complete in case of prokaryotes, and that is inclusion bodies. Yes, come on. Yes, Ankit. So, they are non-membranous stored reserve material seen freely in cytoplasm of prokaryotic cells. What are these inclusion bodies? Yes, this internal structure is known as inclusion bodies. Oh my god. Look here, if you are gonna see in that this violet colored structure is known as inclusion bodies, and yes, and what is their function? What is their function? In case of prokaryotes, the place where reserved food material or the reserved food is stored, reserved food is stored is in case of, I mean, is where inclusion bodies. Yes, inclusion bodies are the places where for reserved food materials are stored in case of prokaryotes. Yes, reserved food materials are stored in inclusion bodies in case of prokaryotes. Example, phosphate granules, cyanophysin granules, glycogen granules, gas vacuoles. All these are examples for inclusion bodies. Yes, that's all are examples for inclusion bodies. Yes, then gas vacuoles are found in blue-green and purple-green photosynthetic bacteria. Blue-green algae, purple bacteria, sulfur, green sulfur bacteria. Or in all these cases of gas vacuoles are the inclusion bodies present. And gas vacuoles, their function is buoyancy. That is helps in floating, right? Yes, that's all about inclusion bodies. So hope that's fine. So that we can move with eukaryotic cells. Yes. So let's start our session with eukaryotic cells. So we have a lot to cover today. 1, 2, 3, 4, 5, 6, 7, 8, 9. 10, 11, 11 more slides to cover today. Yes, eukaryotic cells. They have organized membrane-bound nucleus and organelles. That is what makes difference between prokaryotes and eukaryotes. Membrane presence of membrane bound nucleus in organelles. Yes, presence of membrane gives clear compartmentalization of cytoplasm. They have complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosome. All these are just basic introduction in case of prokaryotes. Only point that you have to add is organized membrane bound nucleus and organelles. Genetic material is organized into chromosomes. Yes, everyone. So the cell organelles that we have to cover in case of eukaryotes are cell membrane, cell wall. These two are the organelles that we are going to cover today. In the next class, it's hope to complete endomembrane system, and mitochondria. Yes, endomembrane system means a system comprising of endoplasmic reticulum, Golgi apparatus, lysosomes, and the vacuoles. Then the very next session it's planned to complete the entire topic, but I don't know whether it will happen or not. Sometimes it will be till plastids to cilia or in flagella, then centrosomes, centrioles, and nucleus in next session. So it makes 3 further sessions more, right? 1, 2, and 3 further sessions more. We have completed 2, total 5, right? In 5 or 4 lectures we will complete it, and we haven't divided till now how to complete it. Yes, Paki. Yes. So we are going to start cell membrane. The very basic introductions, all that similar in case of prokaryotes, we have learned there, and no issues. We have completed prokaryotes, and we are going to start eukaryotes only. So you will be able to cover cell membranes and cell wall in case of eukaryotes. Yes. So keep following with eukaryotes and prokaryotes. You may watch it after the session ends. Yes, Ankit and Sania. So, cell membranes chemical studies on human RBCs show that cell membrane is composed of lipid bilayer, protein, and carbohydrate. Lipid bilayer, protein, and carbohydrate are the 3 major components of cell membrane. Lipids, mainly phosphoglycerides, have an outer polar head and an inner hydrophobic tail. So the nonpolar tail of saturated hydrocarbon is protected from the aqueous environment. Yes. So, have you understood anything? Have you all understood anything reading after reading this about cell membrane? Yes. Cell membrane, also known as plasma membrane. Yes. Ankit meant that you have understood or not? Yes. Okay, fine. Binay, Calvin, and Paki, we will do it. So have stated what took this white screen in order to explain that from my part. Okay. So other terms are plasmolemma. All these are the other terms or other names of cell membrane. Plasmolemma. Then the new name or the latest name discovered or given for cell membrane is biomembrane. Yes. These are the 3 different terms for cell membranes. Cell membrane, plasma membrane, plasmolemma, and biomembrane. Yes. And we have said that this what this cell membrane is composed of a lipid bilayer. Oh my god. Okay. Lipid bilayer. So what do you mean by this lipid bilayer? Lipid bilayer means what? two layer of lipids. Yes, two layer of lipids. And we said that it has a polar head, outer polar head, and an inner hydrophobic tail. Right? It has an outer polar head and an inner hydrophobic tail. What is that? So this is the polar head of this what lipid bilayer, and okay, all these are interconnected. Okay, like this, a circle and two lines. So this is what is meant as polar head, and this is known as hydrophobic tail. In the same way, we have here too. Yes, all these are what tightly bonded. Okay, tightly bonded. And in between you have proteins. Yes, we have to learn the name of all those proteins. Oh my god, one second, it takes time, please wait guys. Okay, oh my god, I don't know wherever I'm going drawing this polar head and hydrophobic tail. Then it consists of another set of proteins. Yes, another set of proteins, and again this lipid bilayer, polar head and hydrophobic tail. Yes, this is all about the lipid bilayer. Two layer of lipids. Lipid bilayer, two layer of lipids. Yes, and you have here in between certain structures, and this is what meant as cholesterol. Between the lipid bilayer, you have what? Cholesterol. Yes, cholesterol. Then this protein, there are two types of proteins, and this proteins is known as peripheral proteins or extrinsic proteins. Extrinsic or peripheral proteins, and this long inner proteins is known as integral proteins or intrinsic proteins. Yes, everyone. Yes. So that's about lipid bilayer and extrinsic protein and intrinsic protein. So hope it's fine. Yes. So lipids, mainly phosphoglyceride, may have an outer polar head. If you look here, you can see an as an outer polar head and an inner hydrophobic tail. Okay. Then ratio of proteins and lipids varies in different cells. In human RBC, it is 52% proteins and 40% lipids. Then based on the ease of extraction of membrane proteins, it's of two types: integral proteins and peripheral proteins. Integral proteins partially buried or totally buried inside the membrane. Look here, partially buried, very sorry, partially buried or totally buried inside this lipid bilayer, we can say that it is integral protein. And look here, here it is in case of peripheral protein, that is lie on the surface of the membrane, that is peripheral protein. Yes, everyone. Hope it's clear in case of peripheral protein and integral protein too, and cell membrane. Yes. Then, fluid mosaic model of cell membrane. It was proposed by Singer and Nicholson in 1972. Then, what is this fluid mosaic model? Fluid mosaic model. According to this fluid mosaic theory, proposed by Singer and Nicholson, these are two scientists, Singer and Nicholson. So, this states that the quasi-fluid nature of the lipid enables the lateral movement of proteins within the overall bilayer. That is quasi-fluid of lipid, that is the semi-fluid. Okay, semi-fluid of semi-fluid nature of lipids enables the lateral movement of proteins, that is enables the movement of proteins between this lipid bilayer. Okay, okay, we will do with integral proteins, please wait. So proteins are helped to, I mean, what the quasi-fluid nature, that is semi-fluid nature of this lipids enables the movement, lateral movement of proteins between the lipid bilayer. Yes, so this ability to move within the membrane is measured as its fluidity. So what is fluidity? The ability of these proteins to move within this membrane is known as fluidity. Then integral proteins. Yes, that's what asked by Binaya. So integral proteins, it is partially or totally buried inside a membrane. Okay, it is totally buried inside a membrane, right? Right? It is this what pink colored one is totally buried inside that membrane. That is when the membrane ends, that protein also ends, right? We can just connect it in such a way. Okay. Whereas in case of this what purple colored one, it's only the partial part of it is covered in this by this membrane. Only partial part is covered by this membrane. Partially covered, partially buried, or totally buried, it is inside the membrane. Then it is known as integral proteins, and if it is lying on the surface, then it is known as extrinsic or peripheral proteins. Yes. Next is the functions of cell membrane. Transport of molecules. The membrane is selectively permeable to some molecules present on either side of it. So cell membrane helps in the transport of molecules, and the membrane is selectively permeable, that is allows only certain substance to enter the cell and only certain substances to leave the cell. Due to the fluid nature, plasma membrane can help in cell growth, formation of intercellular junctions, secretion, endocytosis, and cell division. Endocytosis means taken materials inside the cell, and that helps in their further procedures, that is why whether it is digestion or metabolic activities. Okay, that's about the function of cell membrane. Transport of molecules and the fluid nature helps in cell growth, formation of intercellular junctions, secretions, cell divisions, endocytosis, etc. Yes. Next is the types of transport. How much slides more? 1, 2, 3, 4, 5, 6. Total 6 slides, right? Including this. Okay. Types of transport. Transport is divided. The transport is divided into two types, that is passive transport and active transport. All this you have learned, yes, in your previous chapters, previous classes. Yeah, right. And today also Abhisa have taught you about this transport, right? Active transport. I hope so. I think so. So, moving on with passive transport. Movement of molecules across membrane along the concentration gradient, that is from higher concentration to lower concentration without the use of energy. membranes are moving along the concentration gradient, that is from region of higher concentration to that of lower concentration without any help of any energy. That is known as passive transport, and it is of two types, that is simple diffusion and osmosis. Simple diffusion is the movement of solutes across a membrane, and osmosis across a semi-permeable membrane. Osmosis occurs only in the presence of a semi-permeable membrane. Yes. Is it clear about passive transport? Then, facilitated diffusion. Polar molecules cannot pass through the non-polar lipid bilayer, so they require a membrane carrier protein for their transport. That is known as facilitated diffusion. So, we have said that non-polar molecules are passing through the lipid what? I mean, non-polar lipid bilayer. Polar molecules cannot be passed through that, right? So they require a specialized protein for their transport from polar molecules require a specialized protein for the transport from transport along the non-polar lipid bilayer, and that is what is known as facilitated diffusion. Yes, that is they require a special protein. Polar molecules to pass through non-polar lipid bilayer require a specific protein, and that is known as, and that mode of transport with the help of a specialized protein is known as facilitated diffusion. Yes. Then movement of molecules against the concentration gradient with the use of energy, with the requirement of energy is known as what? Active transport. Okay, across the membrane against the concentration gradient, that is from lower to higher concentration with the expenditure of energy, that is known as ATP is utilized, and that is known as active transport. And example, sodium ion pump, potassium ion pump, all these are examples of active transport. Yes. Next is cell wall. Hope there are three more slides, right? Okay. Cell wall is a non-rigid structure found outer to the plasma membrane of fungi and plants. Yes. In the outside the plasma membrane, in case of fungi and plants, we have what? Cell wall, and for cell wall in case of constitution may be different, right? Constitution in different organisms or different plants and organisms, it will be different, right? So in case of algae, it is made up of cellulose, galactans, mannans, minerals like calcium carbonate. In case of other plants, cellulose, hemicellulose, pectins, and proteins. All these are different constitutions of cell wall. All these algae constitution in this algae we will learn in the next chapter, plant kingdom. Chapter 3, that will be soon covered by Abhisar after digestion and absorption. Yes. So, in other plants, it consists of cellulose, hemicellulose, pectins, and proteins. Cellulose, hemicellulose, pectins, and proteins are the other constituent or components that helps in the composition of cell wall. Yes, yes, everyone. Cell wall of a young plant, that is the primary cell wall, is capable of growth. It gradually diminishes as the cell matures, and the secondary wall is formed on the inner side towards the membrane. A middle lamella is a layer which is mainly of calcium pectate. It glues the different neighboring cells together. So, the three layers of a cell wall is, the first one is primary wall. The first layer is primary wall. Then you have the middle lamellae. Middle lamellae. Then you have the secondary wall. Okay, then you have the secondary wall. Primary wall, middle lamellae, and secondary wall. The cell wall and middle lamellae may be different. may be traversed by plasmodesmata, which connect the cytoplasm of cytoplasm of the neighboring cells. Yes. Is it audible right now? Hope it's fine. Yes. So we have only two more slides more. Okay. So we have discussed that the cell wall in case of eukaryotic cell is of three layers. That is, cell wall is of three layers. That is primary cell wall, middle lamellae, and secondary cell wall. Okay. So primary cell wall is present in case of the young plant cell and gradually diminishes as it matures. And middle lamellae is the main layer, mainly composed of calcium packet and glues the neighboring cells together. Cells present layer that it just glues that. That is, it helps together, holds together. Okay. And cell wall and middle lamellae may be traversed by plasmodesmata, which connect the cytoplasm of neighboring cells. Yes, cell wall and middle lamellae. Traveling connections. Okay, between cell wall and middle lamellae is known as what? Plasmodesmata. Look here, cell wall and middle lamellae is connected by plasmodesmata. That is, so substances can be transferred from plasmodesmata to cell wall. Right? Right? So that's about layers of cell wall. Then functions. It gives shape to the cell. It protects the cell from mechanical damage and infection, helps in cell-to-cell interaction, and act as a barrier to undesirable macromolecules. Yes, cell wall is freely permeable in nature and it gives shape and support to the cell, protects from mechanical damage and infections. From any infections or mechanical damage, it is protected by the cell wall and helps in the cell-to-cell interaction, that is transfer of material from one cell to another cell, as we have seen via plasmodesmata connections. Then it acts as a barrier to undesirable macromolecules, that is large molecules. Yes, yes. Is it fine? That's all for today. Yes. So once again, reminding you, if anyone new is watching this, do join the Telegram group where you have the daily session updates, where you can interact with me, where you have daily polls and you can have regular discussion. Yes, and the direct link to join the Telegram group is given there in the description box, and that is t.me/neat_biopoint. Yes, do hit the like button, do share it with your friends, and do subscribe Biopoint, stream the world of bio. And it's me, Signing out from the team by Punch in the world of bio. Bye bye.